I have to say again how exciting it is to be at the MPRINT conference. I've heard about this before, and I've worked with some of the Vanderbilt people, not fully aware of all the people that have been gathering for the past 4 or 5 years to discuss these kind of topics. So I'm really thrilled to be here and take advantage of the opportunity to share some of the work that we do. Today I'm going to talk about my favorite little blood vessel called the Ductus Arteriosus, and I'll give you a little bit of orientation as to what the Ductus Arteriosus is, but the OBs in the room know what the ductus is and want it to stay open in the fetus, and the neonatologists in the room want it to close. We know that the ductus is a prostaglandin sensitive vessel from long histories of studies. The ductus provides a right to left shunt in the fetus so that oxygenated blood from the placenta can reach the fetal circulation. And the way it does that is for all the oxygenated blood to enter through the ductus venosus, cross into the right atrium, and then it has a decision to make whether to go across the patent foramen or down into the right ventricle. Once it's in the right ventricle, you don't want the blood that's freshly oxygenated from the mother to go to the lungs because the lungs are uninflated, so it goes across this very large shunt called the Ductus Arteriosus, outlined in pink. This bypasses the lungs and allows the freshly oxygenated blood to go out to the fetal body before it returns to the placenta to dump waste and pick up oxygen again. At birth, this vessel closes and hopefully permanently remodels, so it isn't a problem on rounds every day for me, but unfortunately that's not true. The ductus fails to close, especially in preterm infants. There is a PDA that happens in term babies where the ductus fails to close in the normal 48 to 72 hour period. This doesn't happen often, around 1 in 2,000 infants, unless there's some old-fashioned problem like congenital rubella syndrome, or if you have a genetic syndrome with PDA, then otherwise a term baby might have a PDA. Unfortunately, in preterm infants, this is much more of a problem. Up to 60% of tiny preterm infants will have a PDA that causes me a daily problem on rounds trying to figure out what to do with it. Treatments for the PDA largely involve NSAIDs, and these come with multiple adverse effects that we have to find the right balance for. What I've shown in the slide picture here is a diagram of the human ductus and its ligamentum arteriosum after it closes. And the same structure exists in mice, all mammals, reptiles, amphibians, crocodiles. Every animal you can think of that's within the mammal family has this. Salamanders, lizards. So the ductus is a common feature. There's even a fish that has a ductus called the lungfish that opens its duct when it needs to be in the water and closes its duct when it lives on land. So this is a fairly universal vessel that's of concern, and we have lots of models to study it with. This diagram just shows a mouse circulatory system where I've injected blue dye. You can see the widely patent ductus here and the constricted ductus forming the ligament, which closes in the mouse in about 4 hours. We learned early on that in the 1970s that the ductus is a prostaglandin sensitive vessel. We kind of first learned in sheep and other animal models that you can give prostaglandins and make it relax, and then quickly after that we learned that prostaglandin inhibitors help it to close. The first report of such an event was in 1969 when a single report came out of a mother who was taking too much salicylates for rheumatoid arthritis, and she was found to have a ductus in her infant that was constricted at birth, but then soon with birth and withdrawal of the drug, it reopened. But that, starting in the 1969 period on, we knew that this was a prostaglandin sensitive vessel. In the 1970s and 80s, when we developed better ultrasound techniques and started looking at what was inside the fetus, we learned that we could image the ductus, and we also learned that these tocolytic meds that mothers receive to slow down their labor could also cross the placenta. Animal studies originally said that it wasn't freely permeable and there was a 5-to-1 or greater ratio in the mother compared to the fetus, but human studies by Ken Moise and other people later showed that the Indomethacin is easily crossed the placenta and affects the ductus. So it was probably the late 1987 through 90 to 93 before it became clear how much and how and what gestation was important. So this report in 1993 kind of codified material from other investigators, and what they basically arrived at is that a 32-week cutoff point in gestation was a period when the fetus before that time, its ductus didn't care. But after 30 to 32 weeks of gestation, OBs no longer wish to give a nonsteroidal anti-inflammatory as a tocolytic because the fetal ductus can constrict in response to that. So this was the first report, and this paper by Steve Vermillion and others again reinforced this. So we know now that there's a break point in gestation where after a certain time point of development, the fetal ductus is held open by prostaglandins, and if you give a tocolytic NSAID to stop labor, then it will cross and constrict the ductus. So there's a little bit of a conundrum because prostaglandins make the uterus constrict. Depends on the receptor that's in the smooth muscle. So the EP1 and EP3 receptors are contractile and the EP2 and EP4 are dilatory. So you can give prostaglandins to uterine tissue and it contracts. But if you give prostaglandins to a ductus, it dilates. So there's a little bit of a difference in the smooth muscle bed. Interestingly, there was a report that came out in 1993 by Mary Norton and colleagues, along with Ron Kleiman, who's a PDA specialist, showing that women who got Indomethacin during their pregnancy as a tocolytic sometimes had infants who were after birth had a PDA that wouldn't close. So this was opposite of what was expected. We spent a decade learning the time point and the event that mothers getting NSAIDs had fetal constriction. Now we're showing that the newborn, instead of being born with a closed ductus, they're born with a ductus that won't close. This was a one-off observation for a few years until it was repeated again by Kathy Hammerman, who's a ductus researcher in 1998. She also said that tocolytic indomethacin can cause a baby to have a PDA that won't close. So this is a very unexpected finding. You get dilation of the duct instead of constriction. And this has given rise to this concept of paradoxical ductus arteriosus. So how could it be that one drug causes fetal ductus constriction but could also cause patency after birth? This phenomenon led us to do studies in mice to kind of look at the same thing. We weren't purposely starting out to study this, but we were busy looking at prostaglandins in the ductus using the mouse as a model. So the cyclooxygenase genes weren't cloned and identified until the late 1980s. In 1992, we found out there's a COX-2. So that doesn't seem very long ago to me, 1992, Sudeep, but maybe that seems like a long time ago to other people. But so we just recently learned in the 1990s what the COX genes were. Soon thereafter, quickly, people that cloned the genes also learned how to knock those genes out. So the COX-1 knockout gene doesn't go into labor. The COX-2 knockout animal can't get pregnant. There's a fertility problem. So we figured out a way to make a compound heterozygote where the COX-1 gene is knocked out and the COX-2 gene is heterozygous. And we cross males and females of the same genotype, and you get an animal that can deliver because you do a C-section, and 1/4 of those pups would be expected to have the COX-1, COX-2 double knockout. And reliably, we know this is true. So I have to tell a little lab story for a minute because I was working in a prostaglandin biology lab, and we were working on COX-2 problems with implantation. And we acquired the COX-1 mouse, and it was boring to my mentor, so he allowed me to work on it a little bit because I was a neonatologist, and I think about parturition. And we decided in my little lab group meeting that we would make a double cross, COX-1 and COX-2, and make a double knockout mouse. And my boss said, no, that's a dumb idea. You're not going to do that. You have to have prostaglandins to be an embryo. Of course, prostaglandins are essential for almost every step in fetal development. And so, like any good scientist, we did it behind his back. And what we found was that the animals that were this genotype managed to get pregnant. And our very first female, when my lab manager came and told me she was actually pregnant, we were so excited. We came in the next morning when she was supposed to deliver, and there'd been an accident in the animal room where the lixit on the cage stuck open and it flooded the cage. So the animals, where they get their water supply, they play with the valve sometimes, and so the animal was dead. The mother was dead, but she'd already delivered, and all the little pups were floating in the cage. Fortunately, she didn't euthanize or put them in the garbage where the euthanized animals go. She genotyped each one of them, and we found a couple of COX-1, COX-2 double knockout offspring, proving that you can develop all the way through mouse gestation at least and not need prostaglandins. And later we were more successful at making a breeding colony, and we can repeatedly see that these COX-1 to COX-2 double knockout mice are born, but they also die and have a PDA. So this is a genotype, the genotype of a COX-1, COX-2 double knockout animal, Here's this wide open ductus arteriosus compared to the ductus that's supposed to be closed at about 4 hours of age. And we know these were really COX-1, COX-2 double knockout mice because when we do mass spec of their tissues, their ground up organs, we can see that the wild type animals with red bars have the expected prostacyclin distribution. We just used prostacyclin as an example, but the knockout animals had none, no detectable prostacyclin in any of their tissues. So we know this is really a COX-1, COX-2 double knockout. So now we had an animal model of a PDA that's missing prostaglandins. That kind of was a replicate of what was happening to these women that got a drug that suppressed prostaglandins. About the same time, another report came along about the EP4 receptor. So prostaglandin E from the COX enzymes acts on one of 3 receptors. The EP4 receptor, when you knock it out, you get also an animal that has a PDA. And here's these feeble mice that have, they're born, they're fully intact, they just don't breathe very well. They go into probably congestive heart failure as best as we can tell, and they die within the first 24 hours. And this looks like the same picture as last time, but this is a different animal. This is an EP4 knockout. And again, it has really a giant PDA. So they die within a day or so. And we know that they always have a PDA when they die. So that was great. Now we had 2 tools in our hand. We had the enzymes that make the ligand, the prostaglandin, or we have the enzymes, or we have a mouse knockout for the receptor, the EP4 receptor, and it has a PDA. And finally, we wanted to test our theory that drugs could do this instead of just having it knocked out. And so we made an animal model using chronic COX inhibitors. We had a separate COX-1 inhibitor and a separate COX-2 inhibitor, and we gave them separately. We gave them over different days of gestation. We gave some of the drugs combined together, so it's like Indomethacin on different days of gestation. And we gave some chronically over longer periods and looked at the outcome. And this is just a slice histology section showing a closed ductus when it's supposed to be closed, and a wide open ductus when it's not supposed to be, or when it's supposed to be closed but it's open. And so we scored these animals, and we found that the drug inhibited animals had a PDA just like the knockout. And this raised the possibility to us of an interesting hypothesis that I've spent a lot of time thinking about is maybe prostaglandins don't just act in an autocrine or paracrine fashion. Maybe they act in an endocrine fashion, and maybe they act as a developmental regulator instead of just a vasoactive modulator of tone. Because we know that giving prostaglandin E relaxes blood vessels. And so maybe this is different. Maybe you need to have exposure to prostaglandins all the way through gestation in order for the ductus to learn how to be a ductus. So here's a schematic diagram of what that means. This paradoxical PDA is based on the idea that acutely inhibiting prostaglandin production with an NSAID should lead to constriction. And if you do that on any of these later days of gestation and then deliver the animal and looked at its ductus, it's constricted and closed. But if you take away prostaglandins all the way through gestation by inhibiting or deleting the enzymes, you get an animal born at term with a PDA. And if you take away the receptor, which is knocked out all the way through gestation, you get an animal with a PDA. So what that suggests is that with the exact same drug or with a knockout, you get chronic inhibition of prostaglandin signaling, you block constriction. So could this be our developmental program of interest here? So the way we've gone about trying to answer this question is to first look at which receptors are expressed where. And so we take the mouse ductus out, we compare it to the aorta, and you can see in this upper left-hand panel, the prostaglandin E receptors are listed here. The EP4 receptor with advancing gestation, the mouse delivers on the evening of day 19. When all these days of gestation, the EP4 receptor far out expresses all the other receptors. By in situ hybridization, you can see here the pulmonary artery. Here's the aortic arch coming up and around. The ductus connects the pulmonary artery to the aorta. This is a branch artery here. You can see by in situ hybridization, there's really strong localization of the EP4 gene in the ductus wall. So now we know when the EP4 gene is on and it's most predominant, and we know where. It's in the muscle and the endothelium. Pharmacologically, then, we want to test the drugs that are important to test in women. So we learned how to take the vessel out. So we can excise the ductus arteriosus of a fetal mouse, mount it on glass pipettes, inflate it with the right amount of mean arterial pressure that a newborn mouse would have, and we study it using techniques called vascular myography. So this is pressure myography, so I mount it on tips, we pressurize it, and the y-axis shows the diameter of the vessel, x-axis is time. So we get the vessel to an equilibrated state and pre-constrict it with oxygen. So exposure to a small amount of oxygen dramatically lessens the diameter and constricts the vessel. And then we add increasing amounts of PGE2. And you can see the vessel grandly dilates with increasing amounts, even as low as 10 to the minus 12 or 10 to the minus 11th molar PGE2. So this is the concentration response curve. But if we put in the bath an EP4 receptor antagonist called L161, then we can dramatically block the dilation effect. So this whole slide basically just shows it's the right receptor in the right place, and its functions is what we thought it was. It's a strong dilator and it's inhibitable. So to ask our acute versus chronic question, now I have, instead of a COX inhibitor, I have an EP4 inhibitor. And we look to see functionally now if that's also true on in vivo. So I showed you the ex vivo ductus. This is in vivo in a mouse. Here's the wide open ductus. Here's the constricted ductus. And if I give it a pharmacologic inhibitor that's selective for EP1 or 2 or 3, it doesn't do anything. On the x-axis is 100% patency versus 0% patency. And all of these animals had a completely patent ductus in utero. I'm giving this drug to the mother and looking to see its effects on the fetus. The vehicle is the same. This drug, this L161 compound that blocks EP4, gave a mixed distribution of vessels that were partially constricted or fully constricted. And a second drug that we tried, this Ono compound, was completely dependable for closing the ductus in utero. So we shifted to using this drug for our chronic studies. So now acute inhibition at term gestation causes fetal ductus constriction, but does chronic inhibition do the same thing as chronic COX inhibitors? And that's what these studies were for. So if we chronically expose pups to the AE3028 compound, and we do it in 3 temporal windows during gestation, during days 11, 12, and 13, a 3-day period where the dam got this drug, and then we let her stay pregnant and looked at the outcome at term, we'd found no ductus-specific effects, even though there was fairly high mortality rate at birth. About half of the animals died, but they weren't related to anything we could see wrong with the ductus. In the middle panel, in the middle window, there was no ductus effects, and most of the animals survived, so their ductus closed fine. But when we gave that same drug chronically over a 3-day period toward the end of gestation, all the animals survived, but all the animals also had a widely patent PDA. So now we've used pharmacologic measures instead of knockout measures to pick the window of susceptibility when the ductus cares about prostaglandins, and it's late gestation like it is in women. I'm not going to show the studies, but we also did studies looking at L-name exposure, which blocks nitric oxide signaling. So the fetal ductus is held open by nitric oxide and prostaglandins. But what this shows is that nitric oxide is important in early gestation, and prostaglandins are important in late gestation. So you can't just give 2 inhibitors to make the ductus close more. And this just plots out our data in a single plot, that nitric oxide effects are really gone by the last 3 days of pregnancy. And prostaglandin effects become important, and that's when the receptor shows up as well in the end of pregnancy. So now I'm going to focus back on this EP4 knockout animal, because that's kind of a prize for us to have an animal model of a PDA. And if I take the knockout vessels out and I mount them in a myography chamber like I showed you and look at their diameter, and then I compare knockout vessels to wild type vessels, and we expose them to something to make the ductus constrict, KCl, makes it constrict. U4669 is a prostaglandin receptor stimulus that makes vessels constrict. And we compared what happens to knockout and wild type. We saw that the knockout vessel just doesn't act quite the same. Not surprising, because it's a PDA. It doesn't have myogenic tone, so when I add pressure to it, a wild type vessel squeezes back and regains its tone. But when I do that to a knockout animal, it stays flat. And this is a cumulative plot of that effect. When we look at the response to pressure, a pressure diameter curve, the knockout's always a bigger, floppier vessel. Knockout's always in red. The KCl response is blunted. The U46619 response is blunted. And the response to nitric oxide is accentuated. So this makes me think that the ductus is acting more immature. It's big and floppy. It doesn't respond as well like a preterm vessel would. And I told you that preterm vessels are more sensitive to nitric oxide. So we had another bet in the lab, and I lose a lot of money in the lab to my teammates. But we bet that the problem with the EP4 knockout was it just wouldn't respond to oxygen. Because the most important thing that happens when you're born to get the ductus to close is you breathe oxygen and you take away prostaglandins when you cut the cord and let the baby breathe. So reducing prostaglandins and increasing oxygen tension makes the ductus close. Well, I lost this bet too. When you look at the response curve of knockout and wild type ductuses, they have the exact overlappable response to oxygen. And this is sample tracings of a knockout on top and a wild type on bottom. We just warm them up, pressurize them, hit them with KCl to make them respond. And you can see there's a stair-step response down to oxygen. The big difference is the EP4 knockout always has this wavy baseline. And we learned through our colleagues who work on chloride channels that this is called vasomotion. And when we study the preterm ductus versus the term ductus, the preterm ductus always has vasomotion, a little repetitive recyclic signaling that's an innate principle that tiny blood vessels use to auto-regulate themselves. So this is a feature of a premature vessel. So based on that, we looked at maturity markers, and by PCR, we saw that preterm ductus looks like knockout ductus when we compare a few marker genes that indicate maturity. So we think the answer to some of this question is the knockout vessel acts like an immature vessel. But other people have started to study this EP4 knockout animal too, and they did something that we should have done, which is do RNA-seq and look at lots of different genes or look at lists of candidate genes. And so this group's approach was just to look at EP4 knockout versus wild type and compare it to aorta, instead of aortas in the gray bars here. So instead of comparing it to a preterm ductus, they compared it to the aorta and said there's a panel of genes that make the ductus look like it's aortified. It just acts like a big floppy vessel, not a ductus. So the take-home message from all this is there are knockout animals that are informative for what happens when you give COX inhibitors, just like women get, or when you knock out the genes in the COX pathway that give you an animal with a PDA phenotype. So I'm going to shift my talk a little bit and talk about other things that we've been doing in the lab. And one of the things that we'd like to do is not just know about prostaglandin signaling pathways and what makes the ductus different than other vessels. It's let's look at all the genes we can find. What makes the ductus that little stumpy vessel? Why does it act so different than all the other vessels around it? How does it close at birth? We don't really know how it closes at birth yet. So the field is far behind other vascular biology fields. So one of the things that we would like to do is just look at all the genes that are expressed in the ductus. And several different groups have tried to do that. And then in collaboration with Prince, Ken and Carroll, Sarah Van Driesten, and Tamara Lewis, now in Canada, we decided just to compile as much information as we could find. And I'm not going to go through all the figures, but one of the figures that was put together by my colleague Elaine Shelton was just putting together lists of genes and then organizing them in some attackable fashion. And so she decided to list all the key genes that she found that are enriched in the ductus in expression studies and compare them to all the genes that are linked to PDA or to the ductus function. And we found this sweet spot in the middle where there's genes that are ion channels, enzymes, G protein coupled receptors that land in the druggable genome. So now we're starting to get a little bit better idea what makes this vessel tick. And can we approach this pharmacologically with something besides a COX inhibitor? Please, give me something on rounds that I can do besides a COX inhibitor. And the overriding question is, are there ductus-specific genes that regulate fetal or postnatal ductus? During COVID, I put my grad student to work because we couldn't use as many animal models. And so Michael got busy and cataloged every knockout mouse model that has a PDA. At the time we published this, there was 28. Now there's about 33 different mouse models that have a PDA. We categorized them into different groups. And based on some of the talks I've heard over the past day or so, there's some people's favorite genes are up here: prostaglandin transporter, prostaglandin degradative enzyme. We categorize these into prostaglandin genes and other categories of interest. At the same time he was getting this nice list of mouse knockout genes together, I asked him to look at all the human syndromes. Are there any human knockouts? Well, there's no human knockouts, but there are human single gene syndromes. And around 224 of those human single gene syndromes have a PDA phenotype. We looked at 5 different, we looked at OMEM first, and we compared it to DiseaseGeneNet and lots of other, and 5 other data warehouses for gene syndromes. And we found 224 genes. And if you look, there's a red star wherever there's correlation between the mouse knockouts and the human knockouts. So there is about 50% correlation in the mouse list with the human list. So now we're getting a better set of genes that make us think we're getting a handle on what genetically defines the ductus and which among those things are targetable. Moving a little bit more towards the MPRINT theme, we've just started to scratch the surface of pharmacogenomics. We're just now getting our handle on the genetics of the ductus. We're not quite ready to figure out the pharmacogenomics, but again, with Sarah Van Driest and Prince and Tamara as well, we put together this work trying to figure out, is there a pharmacogenomic story in the ductus? And so Prince had the idea and Sarah had the idea to look at a few specific genes of interest along with all other factors that contribute to PDA. And I'm just going to give you a brief look at the last two things we've done to kind of dip our toe into the pharmacogenomic world. But based on all the factors that we looked at, it looked like for babies who were resistant to Indomethacin treatment, in other words, those treatment failures, gestational age and postnatal age didn't play as big a role. But surprisingly, surfactant exposure had something to do with why the ductus didn't respond to Indomethacin. That's a whole separate story we're just now starting to unravel. And the CYP2C9*2 variant looked like it correlated with resistance to Indomethacin response. And if you go in the literature and you look for other papers on pharmacogenomics in the ductus, there's really just a half a dozen. And one of them after this paper came along and said, well, we think it's CYP29C*3, and it favors a better response to ibuprofen. And the most recent paper that came out said, we don't think either one of those is important. It's actually a different CYP enzyme. So the field is ripe for discovery and pharmacogenomic investigation. Falling back to this response to Indomethacin, we're also still figuring out the things that determine elimination. So we've known since the 1980s that preterm babies handle Indomethacin different than bigger babies by weight or by gestation. Our institution is one of the institutions that has used one dose of Indomethacin at birth to try to prophylactically make the duct go away so I don't have to talk about it on rounds. Other people use 3 doses, and even back in the 1980s, Summer Yaffe and his colleagues plotted out the response to a 0.2 milligram per kilogram dose. And you can see from my blue arrows here, if you're a 2-day-old infant that's less than 30 weeks, you don't eliminate Indomethacin very well. And it may be that you're beyond the therapeutic window by the time you get to a later dose. So you may not need to give 3 doses. So our institution has been trying to focus on just giving 1 dose based on studies like this. And more recently, Tamara Lewis and her colleagues, when she was at Children's Mercy, published another more modern attempt to kind of look at pharmacokinetics of Indomethacin in preterm neonates. This is a small number of small babies. So it gives us a good study to step forwards on. We also have worked with Tamara to kind of do other studies. This is a group of 30 infants that are less than 1,000 grams from my institution. And we use a microsampling technique that Tamara taught us and used her lab at Children's Mercy to just look at what happens when you give 1 dose to a tiny baby and watch its elimination. And we haven't finished all the analysis of this, but it's still a project that's underway. And some of that data did contribute to this paper where we looked at effect response ratios, and Tamara is trying to teach me more about model-informed drug development. I'm still trying to learn, Tamara, so I need to come to MPRINT more often. But that's just a smattering of information that we have from the Ductus Arteriosus world and maternal fetal medicines that may affect the ductus. So I'll just close by saying we've spent a lot of time trying to find pathways and mechanisms that make the ductus close or hold it open and vasodilate it as a fetus. And we know that a lot of these things change over time. So you can't just map out these pathways without accounting for whether it's a preterm or term fetus or whether it's a newborn and all the things that happen after birth. So I'll close with that, and hopefully we'll get some questions afterwards. Thank you.